This module describes the importance of downstream processing and the various strategies involved in downstream processing. Any industrial fermentation will comprise of both upstream and downstream processing stages. Upstream processing involves all factors and processes leading to and including the fermentation and consists of three main areas: the producer organism, the medium and the fermentation process. In most cases, this means recovery of a product from a dilute aqueous solution. The primary objective in industrial fermentation is to recover the product efficiently, reproducibly and safely to its required specification while achieving maximum product yield at minimum recovery cost. Hence, this module will introduce to you the entire processes involved in downstream processing following the fermentation. After completing the module, you will be able to recover the extracellular product by using separation techniques, extract the product from the harvested broth, understand the purification methods to recover the pure product used in food industry. What is downstream processing? The product extraction and purification steps carried out subsequent to product formation is called downstream processing. It is the most important part that decides the quality of the desired product obtained from the initial raw materials. The different steps and unit processes used in downstream processing of biotech products highly depends on the nature of the product, the level of purification required and whether the product is intracellular or extracellular. The first stage in the recovery of an extracellular product is the removal of large solid particles and microbial cells, usually by centrifugation or filtration. In the second stage, the broth is fractionated or extracted into major fractions using ultrafiltration, reverse osmosis, adsorption or ion exchange, gel filtration or affinity chromatography, liquid-liquid extraction, two-phase aqueous extraction or precipitation. Afterwards, the product containing fraction is purified by fractional precipitation. Further, more precise chromatographic techniques and crystallization to obtain a product which is highly concentrated and essentially free from impurities. Now, let us move on to the steps involved in downstream processing of a fermentation process. There are 6 important steps in downstream processing of a fermentation process. They are as follows: 1. Solid liquid separation. 2nd, cell disruption. 3rd, extraction of the product. 4th, concentration of the recovered product. 5th step, purification, and the last step is drying. This diagram gives the entire schema of upstream and downstream processing. It gives a clear picture starting from the isolation of microorganisms followed by improvement of the strains. Improved strains enter into the fermentation processes with the selective media in a specific bioreactor. Afterwards, they enter into the downstream processing for the product recovery. Now, we shall discuss the very first step in downstream processing, that is solid liquid separation. In this step, the cells are separated from the fermentation broth. This can be done by filtration, centrifugation, sedimentation, flocculation or gravity settling. Each of these processes has unique principle by which it works to separate the solid content from liquid broth. Let us discuss the first technique involved in solid liquid separation, that is filtration. Filtration is one of the most common processes used at all scales of operation to separate suspended particles from a liquid or a gas using a porous medium which retains the particles but allows the liquid or gas to pass through. It is common practice to use filter aids when filtering bacteria or other fine or gelatinous suspensions which prove slow to filter or partially block a filter. Kieselguhr, which is a diatomaceous earth, is the mostly wide used material. In case of microbial biomass production and when the product is intracellular, filter aids cannot be used. A filter medium constitutes the separating agent and retains the particles according to size while allowing the passage of the liquid through the filter. In cake filtration, the particles are retained as a cake on the filter medium. The flow through the filter layers is dependent on area of the filter and flow resistance provided by the filter medium and the cake. If the particles do not penetrate the filter medium, then its flow resistance will remain unchanged. It is used for the separation of filamentous fungi and filamentous bacteria. Filtration uses pressure created by overpressure or vacuum, and its rate depends on filter area, fluid viscosity and the resistance generated by filter cake, which increases with time. The technique of filtration employed uses various filters as follows: batch filters, continuous filters and cross-flow filtration. In batch filter, a plate and frame filter is a pressure filter in which the simplest form consists of plates and frames arranged alternately. The plates are covered with filter cloths or filter pads. This type of filter is most suitable for fermentation broth with low solid content and low resistance to filtration. Pressure leaf filters incorporate a number of leaves, each consisting of a metal framework of grooved plates which is covered with a fine wire mesh or occasionally a filter cloth and often pre-coated with a layer of cellulose fibers. There are three common types of pressure leaf filters. They are vertical metal leaf filter, horizontal metal leaf filter, stacked disc filter. The next type of filter is continuous filter. An example is rotary vacuum filters. Rotary drum vacuum filters are the most commonly used. The filter is in form of a rotating drum with a partial vacuum on the inside of the drum. A portion of the drum rotates through the medium and the cells are sucked to form a coating on the drum. The cells are continuously scraped off to prevent blocking of the filter. A number of rotatory vacuum drum filters are manufactured which differ in the mechanism of cake discharge from the drum, namely a type of rotary drum vacuum filter with string discharge, a type of rotary drum vacuum filter with scraper discharge, a type of rotary drum vacuum filter with scraper discharge with pre-coating of the drum. Now, the next important filtration technique is cross-flow filtration. For large-scale industrial membrane filtration, cross-flow or tangential flow filtration is commonly employed. The flow of medium to be filtered is tangential to the membrane and no filter cake builds upon the membrane. The advantages of cross-flow filtration are: it performs a very efficient separation, that is, cell retention is greater than 99.9%. It is a closed system, very essential for the containment of organism with no aerosol formation. The process of separation is independent of cell and media densities in contrast to centrifugation. Filter aids are not added. The next technique in solid liquid separation is centrifugation. In centrifugation, the removal of solids relies on the density difference between the particles to be separated and the surrounding medium. Although centrifuge may be expensive when compared with a filter, it may be essential when filtration is slow and difficult. The cells or other suspended matter must be obtained free of filter aids. Continuous separation to a high standards of hygiene is required. Non-continuous centrifuges are of extremely limited capacity and therefore not suitable for large-scale separation. The centrifuges used in harvesting fermentation broths are all operated on continuous or semi-continuous basis. In addition, equipment cost, power consumption, temperature, etc. are the other disadvantages. We shall see about the flocculation and flotation. Small bacterial cells which are difficult to separate even by centrifugation can be recovered by flocculation, that is, sticking together of cells can be induced by inorganic salts, mineral hydrocolloids and organic polyelectrolytes. Since sedimentation rate of a particle increases with size, flocculated cells can be recovered by centrifugation. In cases where flocculation is not effective, very fine gas bubbles can be created by sparging with the release of overpressure or electrolysis. In any case, the gas bubbles adsorb to and surround the cells, raising them to the surface of medium in form of foam, that is flotation. Long chain fatty acids or amines promote stable foam formation. The cells collected in the foam are readily recovered. Flocculation and flotation are used for the most efficient recovery of microbial biomass in some single cell protein production systems. The second important step in downstream processing is cell disruption. Disruption of microbial cells is usually difficult due to their small size, strong cell wall and high osmotic pressure inside the cells. Cell disruption can be achieved by mechanical and non-mechanical means. The disruption process is quantified by monitoring changes in absorbance, particle size, total protein concentration or the activity of a specific enzyme. Several mechanical methods are available for cell disruption. Mechanical cell disruption methods include liquid shear, solid shear, agitation with abrasives, freeze thawing and ultrasonication. Now, we shall consider these mechanical cell disruption methods one by one. Liquid shear has been most widely used in large-scale enzyme purification procedures. High pressure homogenizers used in the processing of milk and other products in the food industry have proved to be very effective for microbial cell disruption. A well-established technique at a laboratory scale is solid shear, where pressure extrusion of frozen microorganisms at around -25 degrees centigrade through a small orifice is performed. Disruption is due to a combination of liquid shear through a narrow orifice and presence of ice crystals. Mechanical cell disruption can also be achieved in a disintegrator containing a series of rotating discs and a charge of small beads. The beads are made of mechanically resistant materials called as abrasives, namely glass, alumina, ceramics and some titanium compounds. Freezing and thawing is yet another technique in mechanical cell disruption. Freezing and thawing of a microbial cell paste will inevitably cause ice crystals to form and their expansion followed by thawing will lead to some subsequent disruption of cells. Let us see the importance of ultrasonication in mechanical cell disruption. It involves cavitation, microscopic bubbles or cavities generated by pressure waves. It is performed by ultrasonic vibrators that produce a high frequency sound with a wave density of approximately 20 kHz. A transducer converts the wave into mechanical oscillations via a titanium probe immersed in the concentrated cells. However, this technique also generates heat which can denature thermolabile proteins. The method can be very effective on a small scale, but a number of serious drawbacks makes it unsuitable for large scale operations. Now, we come to the non-mechanical cell disruption methods. An alternative to mechanical cell disruption is to cause their permeabilization. This can be achieved in several ways: autolysis, osmotic shock, freezing and thawing, or heat shock. In autolysis, the biggest advantage is the low cost and the microbe's own enzymes that catalyzes the process. So, there are no foreign substances introduced into the process. In osmotic shock method, disruption can be achieved by equilibrating cells in buffered sucrose, rapidly harvesting and resuspending in water at 4 degrees centigrade. It is useful for the release of products from the periplasmic space. Several other techniques using various chemicals and enzymes have been developed for small scale applications, but may lead to problems with subsequent purification steps. Organic solvents such as butanol, chloroform and methanol have been used to release enzymes and other substances from microorganisms by creating channels through the cell membrane. Treatment with detergents or alkali can also be effective. Cell wall degrading enzymes such as lysozyme, which degrade beta-1,4-glycosidic linkages, is useful in gram-positive organisms. Addition of EDTA to chelate metal ions improve the effectiveness of lysozyme. EDTA sequesters the divalent cations that stabilize outer membrane proteins. Antibiotics such as penicillin can be used on actively growing bacterial cells in combination with osmotic shock. Now, let us consider the third step in downstream processing, that is extraction of the product. The process of recovering a compound or a group of compounds from a mixture or from cells into a solvent phase is called extraction. Extraction usually involves both separation as well as concentration of the product. There are three types of extraction procedures. First one is the liquid-liquid extraction. It employs two immersible liquids into which the product is differentially soluble. Usually, successfully, smaller volumes of the solvent are used for repeated extraction of a given sample. Back extraction also tends to increase the selectivity of extraction. The extraction may be performed in a single step by multi-stage parallel flow extraction or by counter-current extraction, which is most complex but most effective one. Coming to the whole broth extraction, it should be used wherever possible since it reduces the number of steps as well as product loss. The effectiveness of extraction may, however, be reduced due to the presence of cells. Next type of extraction is the aqueous multiphase extraction, which can be used for separation of enzymes from cells or cell debris. The enzymes are extracted in an aqueous polyethylene glycol dextran mixture which form separate phases. Recovery of enzymes from these phases is rather some of the difficulties encountered in centrifugation. Now we come to the fourth step of downstream processing, that is concentration of the product. Some concentration of the product may occur during the extraction step. Further concentration may be achieved by the following. First one, evaporation. Second, membrane filtration. Third, ion exchange methods. And fourth, adsorption methods. Evaporation is generally used in cases of solvent extraction using various devices. Example, continuous flow evaporators, falling film evaporators, thin film evaporators, centrifugal thin film evaporators and spray dryers. Efficient arrangements must be made for recovery of the evaporated solvent to reduce costs. For low-grade products, often evaporation of the whole broth is undertaken using a spray dryer. In membrane filtration, both concentration and separation of the products can be achieved, usually based on the size of the molecules. The different processes of membrane filtration are as follows: microfiltration, ultrafiltration, reverse osmosis and electrodialysis. Micro and ultrafiltrations work as sieves and separate molecules of different sizes, but reverse osmosis can separate molecules of similar size. Microfiltration can be used for cell separation as well. Let us consider the ion exchange resins now. These are polymers having firmly attached ionizable groups, anions or cations, which ionize under a suitable environment. These may be solid, example dextran, cellulose, polyamine, acrylate, etc. or liquid, example a solvent carrying a functional group like phosphoric acid, mono or diester, etc. Solid ion exchangers may be used in two ways as follows. They may be packed in columns or they may be added to the extract and removed by decantation. Liquid ion exchangers dissolve only in non-aqueous solvent carriers and the separation is similar to liquid-liquid extraction. Some antibiotics are recovered directly from the whole broth using ion exchange resins. The product is recovered from the ion exchangers by ion displacement. This also regenerates the ion exchange. Next is the adsorption resins. These are porous polymers without ionization. Most compounds are adsorbed to the resins in non-ionized state. The porosity of the resin determines the surface available for adsorption. These resins may be apolar, such as styrene divinyl benzene, polar, such as sulfoxide amide, or semipolar, like acrylic ester. The products are recovered from such resins by solvent extraction, change to pH, etc. The final step in the recovery of a product is purification, which aims at obtaining the product in a highly purified state. The earlier steps would have achieved variable degrees of purification, which may determine the degree of resolution necessary during the purification step. The degree of resolution will mainly depend on the similarities of the metabolite with other molecules present in the concentrate and the degree of purity required in the final product. Purification is achieved by the following procedures. crystallization and chromatographic procedures. Crystallization is mainly used for purification of low molecular weight compounds like antibiotics. Example, penicillin G is usually extracted from the fermentation broth in butyl acetate and crystallized by the addition of potassium acetate in ethanolic solution. Crystallization is the final stage in purification of products like citric acid, sodium glutamate, etc. Now, we shall move on to the role of chromatographic methods in purification. These are used for the purification of low molecular weight compounds from mixtures of similar molecules. Example, homologous antibiotics and of macromolecules, especially enzymes which are similar in properties. The materials used for chromatography are generally coated on particulate carriers which are packed in columns through which the liquid containing the product is pumped either upward or downward. The separated product is recovered in some sort of fraction collector. On a large scale, organic solvents are used for collection. Therefore, the whole system has to be installed in a flame-proof and explosion-proof room. The different chromatographic procedures are as follows: adsorption, ion exchange, gel filtration, hydrophobic, affinity, covalent, and partition chromatography. Adsorption chromatography separates molecules due to their differential affinities for the surface of a solid matrix. Example, silica gel, alumina, hydroxyapatite, or an organic polymer. In case of ion exchange chromatography, resins or polysaccharides, example, cellulose, saccharose having attached with ionized functional groups are used for a high resolution separation of macromolecules like proteins. Gel filtration uses molecular sieves composed of neutral cross-linked carriers such as polymers like agarose, dextrons of different pore sizes. Molecules smaller than the pore size enter the carrier and are retained. They are later eluted in order of molecular size and collected. Gel filtration is used in aqueous systems. Hydrophobic carriers are used for purification of hydrophobic molecules. Affinity chromatography uses molecules called effectors to which the product has high and specific affinity. Example, using an antibody, that is effector, for purification of the antigen to which it is specific. The effector is immobilized on a water insoluble carrier which is packed in a column through which the mixture is passed. The effector binds only to the molecules for which it is specific and retains it in the column. It is later recovered by elution using a buffer solution of a specified pH. For example, human leukocyte interferon is recovered in high yield and in high purity by affinity chromatography using monoclonal antibody immobilized on a saccharose column. Group specific affinity chromatography and covalent chromatography are based on chemical interactions between the carrier and the product molecules. The sixth and the last step is drying. Drying makes the product suitable for handling and storage. It should be accomplished with a minimum rise in temperature due to heat sensitivity of most products. Addition of sugars or other stabilizers improves the heat tolerance of some products like enzymes and pharmaceutical preparations. The most common approaches to drying are as follows: vacuum drying, spray drying and freeze drying. In spray drying, the solution or slurry to be dried is atomized by a nozzle or a rotating disc. A current of hot air, that is 150 to 250 degrees centigrade, is passed. The drying is so rapid that the temperature of particles remains very low. Spray drying is used for enzymes, antibiotics and food products. Vacuum drying uses both heat and vacuum for drying. It can be applied both in batch mode like chamber dryers and in continuous mode like rotating drum vacuum dryers. In freeze drying, the liquid to be dried is first frozen and the water is sublimed from the frozen mass. A very low pressure is maintained to promote sublimation of water. The energy needed for sublimation is provided by heated plates and radiation onto the surface. The temperature of solid is regulated by regulating the pressure in the drying chamber. This is the most gentle method of drying and is used for many pharmaceutical products. Example, viruses, vaccines, plasma fractions, enzymes, etc. and in food industries. Thus, the downstream processing plays an important role in the fermentation technology. This module has given the detailed information about the downstream processing. As the products vary greatly in size and nature, different separation techniques are required for their recovery and purification. The liability or sensitivity of many of the bioproducts to the environmental conditions places further demands on the characteristics of the separation processes used for their production. The concentration of the product in the starting material is a major factor in the overall cost of production. Therefore, a holistic approach is required when developing a new industrial purification strategy.